Saturday, fortnightly Saturday ASTI webinar. Today we are pleased to have Professor Neeraj Sinha, who is professor at the Center of Biomedical Research at SGPGI campus. Professor Sinha has done his PhD from Indian Institute of Science, Bangalore. He has been a very prolific researcher in the field of NMR. His research group primarily focuses on methodology developments and application of NMR spectroscopy to understand the intricate biological system and the different inherent biological processes governing human health. The systems he uses also comprises the study of bones and its susceptibility to osteoporotic conditions, gallstone associated with gallbladder cancer, and biomarkers of acute respiratory distress syndrome. So these are really cutting edge, state of the art expertise he has to study biological system. Professor Neeraj has more than 100 publications in international journals, and Professor Sinha, after his PhD, spent more than 3 years, of nearly 4 years, in USA at places like Iowa State University and University of California, San Diego. Professor Sinha has published, as I said, more than 100 papers, and he has written several book chapters, and he is also very well funded by competitive funding mechanisms from extramural sources. So together, we are delighted to have today Professor Sinha to talk about the use of solid-state NMR spectroscopy of bones, which is I'm sure that to this young audience, it would be a new area, but that is the whole purpose, to acquaint ourselves to new areas. And I'm sure Professor Sinha would be explaining to this predominantly biological science background audience, so that it becomes very clear and lucid to understand something which is a highly specialized area to study bones. So he will talk on solid state NMR spectroscopy of bones. And thank you, Professor Sinha, for taking out your time to present this webinar. And now the floor is yours.
Thank you very much, Dr. Naveed, and for inviting me to this interesting meeting of bone biologists. And this is actually a very interesting platform where I would like to show what we can understand by applying solid-state NMR to the bone. So I'm basically a spectroscopist who try to develop different techniques to look at systems. So bone is one such system where we have been interested in solid-state NMR application. So I think I would like to ask for pardon. I will not be talking much about the biology of the bone, because this is something which I don't understand much. But I can tell you what are the information we can extract by applying solid-state NMR of bones. And then definitely in terms of applications, there are many bone disease conditions where people have started using solid-state NMR to understand the bone biology. So please do not expect a lot of biology coming from my talk, but a lot of spectroscopic information and bone detail which we can extract from this talk. So So outline of my talk is first I will try to give introduction to NMR spectroscopy. What do we mean by nuclear magnetic resonance spectroscopy, and especially when we talk about solid state, so how different it is from the routine NMR spectroscopy. And then to the bone structure, like what we need to understand about the bone structure and how this solid state NMR can be applied not just to the bone but to the cartilage, and what are the advantages we can get by this spectroscopic technique. And then I will talk about newly developed method, which is a new thing coming up in NMR field, which is called as dynamic nuclear polarization, and how it can be even further useful for bones. So NMR is a spectroscopic technique, and this is basically deals with the nucleus of the atoms. So most of the element in the periodic table have a nuclear spin, which is a property of the intrinsic property of that nuclei. For example, like hydrogen has a spin half nuclei, and carbon 13 also is a spin half nuclei, but carbon 12 is a spin 0. So I'm talking about carbon 13, which is a very rare isotope, 1.1% is there. And similarly nitrogen 15 and other phosphorus and silicon, they all these nuclei have a spin property, and because of this spin property, when we put inside the magnetic field, very strong magnetic field, their nuclear spin energy level gets aligned either with the parallel with the magnetic field or with opposite to the magnetic field. So whenever we put any sample in the NMR spectrometer, all the nuclei which are NMR active, their spin gets aligned either parallel or non-parallel or anti-parallel with the magnetic field. So this create a small energy difference between the two states, and the difference between these two energy states can be excited by applying a suitable radio frequency pulse, and this is being done by this RF coil, which is matched with the NMR probe inside that, and then we put our sample inside this. So once we excite from lower energy level to higher energy level, definitely we can measure those radiation, and then this radiation can go to the electronic part, and then finally with the appropriate signal processing and detection we can get the NMR signal, and this NMR signal comes from this sample, so definitely it has a signature of chemical structure about this. So in NMR spectroscopy we try to map whatever is the interaction present experienced by the nuclear spins by looking at the NMR spectra, which is being generated by putting the magnetic field. So NMR can be done for two types of system. Either we have a solution, and in solution we get different position, and you can see here that the hydrogen nucleus comes around here, fluorine is here, and then phosphorus, lithium, carbon, and everything is different. So all these different types of nuclei have a different intrinsic frequency. It is because their nuclear properties are different, hydrogen being the highest magnetic field and then the highest frequency into that. So based on the position of this line, we can identify those nucleus, as well as we can also identify many of these structures. So this so far so good, but when we have the sample in a solution state, we get a very sharp line, but we can also do NMR in the solid state. So in the solid state, for example, like this type of molecules are there, and then we are looking at the carbon. So the carbon will see all the interactions of the nearby nuclei, and because of these interactions, their chemical shift or the position in the NMR spectrum are different. Like this kind of carbon comes at 90 ppm, and this kind of aromatic carbon comes around 160 ppm, and then the aliphatic comes around 50 ppm. It is because all these carbon have a different electronic environment, which gives rise to the NMR frequency being different. And then based on these difference in frequency, the techniques have been developed to figure out what are the different molecule present in the sample which has been subjected to the NMR spectrum. Not only that, when you have a solid, you also get a pattern like this, depending upon how is the electronic environment around that. For example, like these three are different types of electronic environment having different. So in solid, you don't get a sharp line, but you get a pattern like this. And these patterns are also characteristic of the molecule which we are looking at. So over the period of time, the technique have been developed that even the sample is in the solid state, we can still get sharp line and identify the molecule. And this is called as magic angle spinning. So this is just a one technique being used by solid state NMR spectroscopy to get a sharp line from the sample, which otherwise gives a broader spectrum. So what we do in this kind of technique is we put our sample inside this rotor, and this rotor is kept at a particular angle which we call as magic angle, 54.7 degree, with respect to the magnetic field. And while recording the NMR spectrum, we spin it very fast. Very fast in the sense that 10 kilohertz, 20 kilohertz, or even the 100 kilohertz technology is there. And while spinning, the NMR spectra is being recorded. So even though the sample is in the solid state, but by employing this technique, we can still get a sharp solution like the spectrum, and it really helps in getting the high resolution spectrum of the NMR signal from the sample which is in the solid state. So in terms of pictures, this is how the NMR spectrometer looks like. These are the two NMR spectrometer in CBMR, and this was funded by DST, and this is the 800 megahertz solution state NMR. So you can see this is the magnet, which is a superconducting magnet, and then we put our sample from the top in a tube, and it goes here, and these are the electronic part by which we excite the signal and then detect it and do signal processing. And finally, the NMR spectra is being recorded here in the computer, and then we get a analysis part of that. This is another spectrometer which is only for the solid state, where we have magnet here, and then all those magic angle spinning technologies are there. Well, the NMR spectrometer can be very big depending upon the field it produces, and this is one such 900 megahertz solid state NMR spectrometer at University of California at San Diego, which was so big that we had to take stairs to reach at the top while putting the sample. But this was a very high field, so that we can get even a very high resolution. So high field has a lot of advantage in going, but the technology also makes it more expensive and complicated into that. So in terms of application, NMR has been developed to such an extent that it has found application in almost all branches of modern science, whether it's a chemistry or pharmaceutical sciences, where whatever molecule that you synthesize can be mapped by the NMR spectra. And then this is a very integral part of chemical chemistry research. And for the biology research, protein structure can be mapped by the NMR spectroscopy. And then for many of these important protein, which has a different binding site and all those things, can be mapped by this application of NMR spectroscopy. It has an application in the material science for geolites and those kind of important polymers, and its NMR spectra can be recorded in order to understand its structure. Even the medical application of MRI is a well-established. MRI is basically a technique of NMR spectroscopy, where we use NMR spectrum to map the integral part of the human body, and then this mapping images are related to the physical state of the organ for which we have produced this image of that. And NMR also has a very lot of application in the petroleum industry to understand, to check the purity of the crude oil and things like that. So let us come back to our own things like bone and cartilage. So I think this audience already know what do we actually mean by bone. So on the macro scale, you have a structure like this, bone tissue, And then if you go to the deeper level on the 100 micrometer diameter, then you have structure, osteon and Harvestian canal. But if you keep on zooming and going to the deeper and deeper level, you know that it is made up of mineralized collagen fibril, and then other part are arranged in a hierarchical structure. So the idea is like if we have a bone with a compromised mechanical property, then if there is any change here, this will be reflected in the macroscopic scale. So that is the idea, like if there is any change in the microscopic part of the bone, it will manifest itself in the bone mechanical property at the macroscopic level. And solid state NMR is the only technique where we can look all those interactions, molecules present here in a very high resolution stage. So that is what has led to a lot of application of solid state NMR to the bone and cartilage. So in terms of what we can see and how the solid state NMR has made a main contribution, and recently we know that BMD tests are no longer a reliable predictor of bone mechanical property or the fracture risk. And this was shown if we just look at bound water volume fraction and pore water volume fraction of a young and old person, then there are significant variations going from here to here. So not only that, the peak stress which a bone sample can attain is also correlated with the bound water volume or pore water volume fraction. So this result shows that the water plays a very important role in the bones, because bones mechanical property not only depend upon BMD and other factors, but it also depends upon how much water is there, which also changes when the patient goes from young to old age. So definitely old age has a weaker mechanical property. Old age bone has a weaker mechanical property. So all these results started the uh, question: how does the water plays an important role inside the bone matrix? Where are the water content, and how it is playing a role in the bone structure, which ultimately lead to the important role in terms of its mechanical property. So what we do is the solid state NMR, we take a bone sample, put inside this rotor, and then do a magic angle spinning and other spectrum to record its spectrum. So in terms of component, bone has inorganic mineral, organic protein, lipid, and water. And then we can almost observe all these components inside the bone matrix. So if we look at different experimental methods, then bone has been subjected to all these spectroscopic methods. But all these spectroscopic methods require bone samples to be processed. For example, like for X-ray, you need some good crystals where you need to do some processing, neutron diffraction, backscattering, wet chemical analysis, or FTIR. But solid state NMR is the only technique where you don't need any processing. What we do is that we take out the bone sample from the animal and just cut a small piece of this and put inside the rotor without any chemical treatment or doing anything with the bone sample. So that is one such advantage. Like if the bone can does not degrade over 3-4 days of time, then whatever we observe from the NMR spectrum of bone sample, it will be a true reflection of bone's composition when it was inside the animal system. So this is a very important aspect, that native bone sample can be studied by solid state NMR. But then there is also a problem, because we cannot preserve bone for a very long time without putting something in a preservative kind of environment. And that also affects the NMR spectrum. So you see, if the technique is very sensitive, then a small perturbation can also change its results. So that is also problematic. So I'll show some of the results where what we got in terms of probing the role of water inside the bone matrix. And this is one such experiment where we try to look into the lipids of the bone matrix. So lipids are not only there in the bone marrow, but they are also in the bone mineralized tissue, but its constitution is about 2%. So major constitution is triglyceride. So by solid state NMR, we can look at the lipid also. Just for an example, I'm showing this is the carbon 13 NMR spectrum of a bone. So carbon 13 means it is the organic part of the bone matrix. So it mainly has a collagen protein, and we can almost assign all the carbon 13 spectrum coming from the collagen protein, including citrate signal can also be seen, although its concentration is very less, but we can still observe them. And hydroxyproline, gamma, and all these resonances comes from the different part of the collagen protein when it is inside the bone matrix. So this is the spectrum of collagen. When collagen is inside the bone and everything, all the interaction and everything is present inside that. And this is the inorganic component. So inorganic has hydroxyapatite. So we can look at the phosphorus 31 signal and how it is interacting with the water component and OH part. All these things can be mapped by that. If we just look at the hydrogen spectrum, then we mainly get signal coming from the water and then coming from the OH part, and the lipid part signals are these small signal which comes from here. So yeah, so almost all components of bone matrix, whether it's organic, inorganic, or water, can be mapped by NMR spectrum. So this gives us a lot of power to look into the other things like how the lipids are there and what is the interaction between water and lipids and water and mineral and water and collagen, and all kind of fancy things can be done. So we have been developing those methods to look at the collagen protein. So collagen is a triple helical protein, and we can generate spectrum coming from the collagen part of that. And these kind of spectrum, which is a two-dimensional spectrum, is a very good indicator of how the collagen protein structure is there. In fact, we have found that if there is a small perturbation in the collagen structure, this will be changing this spectra drastically. So all these spectra is a very good film for what is collagen is happening inside the bone matrix. So let us look at the lipid part. So in the water part, we can differentiate mobile water, bound water, and free water inside the bone matrix because they have a different type of correlation time. And then we can also look into the lipid part of that. In fact, we have done some NMR based method to look into those lipid part and get a signal exactly coming from the lipid, even though water is also present inside this. And this is the kind of spectrum we get from the lipid part. So lipid is mainly triglyceride, and then this is the structure of the lipid, and we can assign almost all different kinds of lipid resonances coming from the bone matrix. Well, this gives us even to probe even further and then look at how this lipid part is present inside the bone matrix, like how it is interacting with different components, including the water content. So we measured the relaxation property of these different part of the lipid by some experiment which is called a CBMR experiment, and all these resonances have a different types of bone will map the local lipid environment into that. So what we did was we tried to map on three different types of bone sample. One was the hydrated bone, and the other one where we have exchanged proton with the deuterium. So if the water is exchanged with the deuterium, D2O, then there will be change in its subtle change in the structure of those lipids. And the other one where we have removed the water content from the bone by doing dehydration. So we know that the water plays a very important role in bone mechanical property. So if you remove the water by doing a dehydration, then what are the parts which gets more affected is something will tell us that is where the mostly water component was there. So based on this analysis, we could figure out that these are the part red one, and these two are the part where there changes in because of HD exchange and dehydration was maximum. So they were the one which was finding a strong bonding with the water content inside when the lipid was inside the bone matrix and giving structural involvement. Well, then other important part of the bone is collagen protein. So collagen is a triple helical structure, and then it has mainly proline and hydroxyproline as important amino acids, and glycine is anyways there. And because of these glycine XY, where XY are proline and hydroxyproline, collagen has a very compact structure, and this is what gives rise to the bone as a strength and tensile properties in the bone matrix. But it is not just that XYR proline and hydroxyproline. There are other amino acids, but its concentration is very less. For example, type 1 collagen has other amino acid. Type 2 also has different types of other amino acids. So one of the question was like why these other amino acids are there in the collagen, because they have a very bulky side chain, and it is not clear that how it forms the triple helical structure inside these when these bulky chains are present into that. So all these things can be proved by solid state NMR, because we can map the carbon 13 and look at those signals coming from the bulky part of that. But one of the problem of solid state NMR is like if we are doing it in a natural abundance, then we get a very poor sensitivity. So some of the typical experiment take 2-3 days or even more than that, because carbon itself has a 1.1 percentage, and then inside the bone there are very few carbon, very few collagen, and among those collagen the aromatic amino acids or side chain amino acids itself are very less. So the experiment takes a long time. So recently this technique of dynamic nuclear polarization has been developed, where we can increase the sensitivity of the solid state NMR experiment. So what they do is that they put a electron biradical, and then they do transfer from polarization from electron to the nucleus, and then observe a high sensitivity of the collagen protein signal. So that is a very promising technique, and we have been trying to develop this method for study of bones and cartilage and how it can give some extra information which is not possible by other spectroscopic technique. So as an example, like if we just do a DNP, then this is the kind of spectrum, carbon spectrum we get from the bone matrix. If we don't do DNP, then in the same time we get almost no signal, and this, so we get an enhancement factor of 30, which significantly reduce the experimental time for the probing the structure of this bone matrix. This is something which we have also tried for the cartilage. Cartilage is a more cartilage has very less collagen, and then there are a lot of water and fluid component into that. And we can also look at the nitrogen part. Nitrogen is the backbone of the collagen, because so it has a very less concentration, and nitrogen 15 natural abundance is very less. So we need to have all these newly developed DNP based technique to get its signal coming from the different part of the collagen and 15. So this is one such new result which we got by applying this DNP, like looking at the aromatic part of the collagen amino acid. So this is the sensitivity enhanced bone spectrum coming from the collagen part of the bone. So we see citrate, we see hydroxyproline, and all those resonances coming from the collagen part. But we also see a lot of signal coming from the aromatic part of the collagen protein. So aromatic has a very less concentration, but it is also signal can be seen because of this dynamic nuclear polarization enhancement. So this led us to look in more careful way, like how these aromatic amino acids are arranged inside the bone matrix. So for this we did experiment where we look at proton or hydrogen correlation with respect to the carbon chemical shift. So you see aromatic resonances are coming here, and then we see something here. This is something where we get a one bond correlation between carbon and hydrogen coming from the aromatic part into that. But if we increase the contact time, which is a parameter in the experiment condition, we can also get a long range correlation coming from different part of that. So if we increase that, we start seeing some signal coming from here, which was not present here. So this shows that this is the aromatic part which is correlating with this part of the collagen protein because it is especially close to that. So if we even though further expand this part, then we can see that the aromatic part is basically correlating with proline, proline beta, and proline C gamma, as well as the hydroxyproline beta. So what this experiment shows is that these aromatic amino acids have a spatial proximity with this part of the other ring coming from the hydroxyproline, and especially coming from the beta proline and all these things. So this is one such example where we could see how the aromatic part in the collagen protein inside the bone matrix is correlating with other part of the collagen protein into that when everything is inside that. So this is one such direct evidence of CH pi interaction which stabilizes the aromatic amino acids inside the bone matrix. Well, we did a lot of other experiment for finding out other amino acids role, and then other part into that, mainly finding out the structure of the collagen protein when it is inside the bone matrix in a very high resolution state. So cartilage also we could identify the signals and coming from the GAX. So cartilage has a GAX molecule which is a flexible molecule and a binding part of collagen with other mineral part into that. So cartilage has a lot of water, but its collagen resonances can be assigned mainly from side chain as well as from the backbone part, including the carbonyl part of that. And this GAX signal can be observed into that. So this led us to understand, design more experiment where how the GAX structure is placed inside the bone cartilage uh, into that. So aromatic amino acids correlation with citrate can be seen, and then other part of the carboxylate with respect to the glycine alpha can be mapped by this kind of experiment. And citrate, how it is interacting with the collagen protein through alanine alpha, arginine beta, glutamine and beta, and alanine beta is something which we could map in this kind of short and long range interaction of bone ECM. In cartilage, GAX is an important molecule because GAX is something which binds with the collagen protein, and then GAX molecular interaction with other part of the collagen protein can be mapped by this kind of experiment. Not only detection, but the other part, how it is interacting inside the collagen matrix. So all these experiment was done when we have a native cartilage without any processing put inside the NMR rotor, and then with the magic angle spinning and DNP we could see everything. So that is the advantage of solid state NMR. Like we can look at the system without any chemical processing. And after the experiment, bones and cartilage are still fresh if it was done on a room temperature, and then further studies or other biophysical studies can be carried out because the sample does not get destroyed. Uh, we apply radio frequency to the nucleus, but nucleus does not have any chemical property, and then when the experiments are over, we don't break the nucleus or the molecule, so everything is intact. So the other advanced experiment of mapping the collagen structure, including the other glutamate, proline, lysine, and its spatial proximity, as well as the carbonyl part of the collagen protein and how it is mapping with the backbone of the collagen protein can be mapped. And then for the cartilage also, this collagen structure is different compared to the bone structure, and this is obvious also because uh, protein structure depends upon what kind of interactions are present inside the system, and definitely cartilage has a different structure. So even though it has the same collagen protein, it has a different structure in terms of these different interaction which we see in the cartilage but do not see in the bone matrix. Backbone of N15 from the cartilage and the bone can also be mapped by these kind of experiment, and for the cartilage it is different in terms of that. So with this, I hope I have convinced that solid state NMR can map some of these interactions, which is not possible by other spectroscopic technique unless you do some chemical processing and change the system. Then of course X-ray can give much higher resolution structure or even the comparable, but in solid state NMR all the interactions are um, present in the full form. So there are some sensitivity enhancement method which is giving a very promising results. But what is the like future? So future is to look at those bone sample which is mainly coming from different disease conditions like osteoporosis, osteogenesis imperfecta, etc. And we have done some experiment on osteoporotic rat bone model and found some marker on in terms of solid state parameter, which we published some time back. But this is something which where the future lies. And very recently, I found one paper which came from the Chinese group, and they use solid state NMR and shows that calcium phosphate formation in a diseased bone is very different uh, from the healthy bone. I still have to go through this paper very carefully, but definitely low bone mass normal and high bone mass calcium signal or the phosphorus signal can be measured, and we can see the changes coming because of these region into that. So with this, thank you very much, and if there are questions, I'll be more than happy to answer.
I think there is a background noise. Thank you very much, Dr. Sinha, for this lucid presentation. I'm sure that the students would be greatly benefited from this presentation. Just that you are aware that we actually had worked together. Dr.
Right.
Sinha and our group has worked together, and we have shown that in osteoporotic bone, the animal model, the total water and particularly the atomic space between the crystal and organic surface area are affected. Um,
Correct.
right? So that we did. We are still looking forward to doing some more studies to understand the bone at the atomic level. So far, we do just micro level, but what he does is at the atomic level to understand the bone material at that condition. And why it is so important, which he has clearly explained, is that you don't do any modification to the bone. You just take the bone and without any fixation, without any kind of chemical or any kind of modification, you are measuring or assessing bone inside too. The way it is there, just that. So that is the best, as close as you can come. So wonderful learning experience also. If students or anybody in the audience has any question or clarification, please feel free. Please ask. If not, then let me again thank Dr. Sinha, and we will be in touch. And you also know all the participant, the audience know his contact. And if you have any idea working with him, collaborating, that is another purpose of this meeting. Get in touch with him and learn more from him.
Sure,
Thank you
sure.
very much. Thank you.
Thank you very much, sir, for giving me opportunity here.
Yeah.
And I look forward to other discussions, future discussion, and future work.
Yes. Yes. And actually, another purpose of this talk is this webinar is that your lab is already in the loop of this email. So um, the next one I think would be on 17th, will be given by Dr. Ashok Kumar at IIT Kanpur.
Okay, okay, sir.
So please, means you will get all these notifications. So tell your lab crew to join, and you also join. And also, if you have any speaker in mind, particularly young researchers ready for giving a talk, please let me know.
Sure, sir.
Sure.
Sure. Okay.
Okay, then. Thank you so much.
Thank you,
Bye
sir.
bye.
Bye.
Bye bye.